So, hi, good morning. Good morning, everybody. Welcome to the CPHMS webinar. Before we begin, I would like to call upon Dr. Suraj, the Senior Research Associate at the Center for Predictive Human Model Systems, to speak for a couple of minutes about the center and the kind of work we carry out here. So, Dr. Suraj, over to you.
Yeah, thanks, Sham. I'll just take like few minutes of your time and just give a brief this thing about what the center is. So thank you everyone for joining today on this Saturday morning. So 1 second. Okay, can you see the slides?
Yeah.
Yeah, we can see. Yeah.
Okay. Like Sham
Yes,
said, we are okay. So like Sham said, we are the Center for Predictive Human Model Systems at Atal Incubation Centre CCMB, and this center was established in 2019 as a collaboration between Atal Incubation Centre CCMB and Humane Society International India. And this was primarily established as a center to enable human relevant research in India. And towards this goal, we have been conducting several activities. So in the past 1.5 years, we have written 3 white papers on various human relevant research, including microphysiological systems, pathway-based research such as adverse outcome pathways, systems and computational biology. And these white papers have tried to understand what are the challenges in terms of funding research infrastructure to conduct this research in India. We also conduct several workshops and webinars, and this webinar is also part of the same initiative to increase more awareness in India amongst researchers and students for these methodologies, which are based on more human biology and not based on animal and not use animal models. So, and we also recently gave out a grant in the field of to build an adverse outcome pathway in the field of cancer last year, and 2 Indian scientists have been awarded that. And apart from this, we also regularly write reviews and popular science articles in journals and various news media outlets, again to increase awareness amongst both researchers and students and general community, scientific community about these methods. With this, I would just a brief intro. I would stop there.
The child is born with this kind of, pseudo adult brain, I should say. And what is surprising is that every hour during this period of 29 days to 52 days, 15 million neurons are born every hour. Okay, that is really, really fast. It's one of the most fastest proliferating organs in the human body. And it is also not very surprising that the brain growth keeps happening until the age of 25. At 25, we have reached the plateau of our brain growth. And then from 25 to 40, 50, we just have this kind of, homeostasis maintained. And after that, again, you have a neurodegeneration. Slowly, these neurons get degenerated. So that's something which normally happens for every human. So, how do we understand how brains develop? So far, what are the tools which are available for us? So normally when we have humans, we run the most common organism we resort to is a mouse model, right? Most of the studies we have are in mouse. Or in fact, flies. I mean, these are really unlikely model organisms that you think of. I mean, imagine that a small fruit fly which comes to a rotten banana or a rotten fruit can actually mimic the workings of a powerful human brain. So the architecture, the way our brains are built are very conserved. And most of what we know about our human brain actually stems from these model organisms. So we owe a big thanks to these 2 model organisms. But if you see a lot of course, you would understand that there are a lot of differences between a human and a mouse and a fly brain. Okay, least of all is the size. So if I were to now just plot the size of a fly versus mouse versus human brain, you can clearly appreciate that flies, I mean, it's really like a speck of mustard, okay, the brain size, even though architecture wise it is primarily conserved. Mouse, again, it's small. And human, of course, we have a bigger brain size, and the startling difference actually starts settling in when you start looking at the way the brain is shaped and the way the brain is folded. So a mouse brain tends to be lissencephalic, which means that it has a smoother brain surface without having a lot of these gyri and the sulci, the foldings that we see in a human brain. So those seem to be absent in the mouse brain, and those could clearly come from those could clearly stem from different types of cells which kind of contribute to this kind of a folding. So we have, apart from having a smaller brain, the fly and mouse, we also have certain specific types of cells and certain specific types of tissue architecture which is missing in these organisms, which in the end we see that could contribute to a lot of diseases that we see in humans are not recapitulated in these organisms. Okay. And then we talk about brain size, right? Because that is something which we all talk about, like humans have bigger brain size compared to our body, we have a bigger brain size. But actually, that is not true. So if you were to imagine an elephant has 7 times a bigger brain size than a human. And now if you then take it to a blue whale, you can imagine how much bigger brain size. It has like 15 to 16 times bigger brain size than humans. So does it mean that just the brain size can contribute to cognition and whatever we see? I'll just give you a scale to show you what exactly I'm referring to. So if you compare a human brain to an elephant brain or to a dolphin brain, you clearly can see a big difference, right? The elephant brain and the dolphin brain are way bigger. Does it mean that they have better cognition? Now we compare this to a gorilla brain, which is a great ape family, right? And they are shown to mimic a human behavior much more closer compared to dolphins or elephants. I mean, of course, in their own ways, they are intelligent. Probably we still don't understand what they are doing. Nevertheless, cognition as we understand it and the brain size are not directly linked. That is something which we have to keep in mind. So if this is the case, then how can we reliably understand the working of a human brain? Okay, so one way to do that is exploring the use of human stem cells. So just like the first video I showed you where a plant emanates from a single seed, like that the whole body for us emanates from a totipotent embryonic stem cell, which we call as the zygote. So you have the fertilized zygote, and then you have the 2-cell, 4-cell, morula, gastrula, all those stages you would have all read about. So those totipotent stem cell can give rise to the whole organism. And the next layer of totipotency actually is marked by pluripotency. So it's plurality. Toti is total, it can give rise to a total organism. Pluripotent is that plural, they can give rise to multiple different types of tissues. For instance, pluripotent embryonic stem cells can give rise to the 3 most commonly known germ lines we know of: endoderm, ectoderm, and the mesoderm. So endoderm is like internal organs, as the name implies. Mesoderm is all your muscles and all that. And ectoderm is your skin, the outer layer skin. And in fact, brain actually comes from the ectodermal lineage. So if you now slowly nudge these cells, these lineages with specific cocktails, they can give rise to multiple stem cells. And finally, you can get differentiated, terminally differentiated cells like the neurons, you can get the skin, and from the mesoderm, you can get all your blood cells, you can get the heart muscles, and from the endoderm, we can get the lungs and the pancreas. So the whole concept of embryonic stem cells actually stemmed when people started using pluripotent embryonic stem cells to push them into one of these lineages and give rise to a particular organ of their interest. So what revolutionized the whole field was the growth of the identification of induced pluripotency. So people discovered that this was not a unidirectional movement where from totipotent you have a pluripotent and then you have multipotent and then you go to terminally differentiated cells. In fact, people showed that even from a differentiated, finally differentiated skin cell, you can go back and make pluripotent stem cells. Okay, this is like literally erasing all the stuff you write on a white paper and then make it into a clean slate. This is what we are talking about here. So these finally differentiated cells, just by using a cocktail of 4 different factors called as Yamanaka factors, can go back to becoming clean slates, and then now you can differentiate them into any one of the other lineages. So that induced pluripotent stem cells is what has revolutionized the whole field of regenerative medicine. And in fact, for a lot of stuff we are going to see with the organoids down in the top, people have used induced pluripotent stem cells. Okay. So the first demonstration of self-organization of cells was way back in 2003 by a pioneer scientist called as Yoshiki Sasai. Unfortunately, he's no longer with us. So he found out that if you take embryonic stem cells and if you just gave them some cocktail of chemicals, they could give rise to self-organized three-dimensional spheroids. Until that point, we were all using typical neural tissues and neural cells, which were all coated on a plastic. Of course, your body is not coated in plastic and your brain is not stuck to a plastic. It is not like a two-dimensional tissue that you're talking of, right? So we have a three-dimensional architecture. And that three-dimensional architecture is what gives us particular cues, spatiotemporal patterning, morphogen gradients. All that happens only when tissues are in three-dimensional, in their three-dimensional form. So he found out that when you have embryonic stem cells with a particular cocktail of factors, you can end up with a beautifully organized tissue, which very closely resembles neural tissues of a human. So 10 years from then, there were further protocols which developed by another very talented lady called Madeline Lancaster. And she came up with this concept of brain organoids, okay? And they clearly showed that these brain organoids that you see in the middle of the picture here in white color, it looks like a white cloud, right? So that is a brain organoid. And by modeling those and by sectioning them, she clearly showed that if you compare a human brain, this is a cortex. So you have three parts of the brain: a forebrain, midbrain, and the hindbrain. So forebrain, we normally have the cerebral cortex, and then you have the midbrain, and then you have the hindbrain, which is your hypothalamus and everything. So she showed that just by for forebrain, she showed that if she sections them, the architecture of human brain and the organoids are very closely linked. So in red color, on the left side for the brain, you can clearly see that you have all the growing neural stem cells in red color, and they give rise to the green colored layer of the neurons that you see there. Okay, so neurons are your basic functional units. So that is how we have all the thought process and everything going on in our brain. And now on the right side, if you look at the organoids, you have a very similar architecture. You have a beautiful layer of the neural stem cells in red color, and then in green color, you have the neurons which have been differentiated from those neural stem cells. So this then started revolutionizing the whole field of how people started understanding, especially early neural development. Okay, so how do we make mini brains in the lab? So we'll start first with embryonic stem cells. So we take embryonic stem cells, they grow as colonies, and then we start making them into embryoid bodies. Okay, so these closely resemble those clumps, beautiful balls of cells, which can now go into any one of the lineages: endoderm, ectoderm, or the mesoderm. So we add a specific cocktail of mixtures which can result it to go into an ectodermal lineage. So you end up forming neuroectoderm, which closely resembles your neural tube. Okay, and then we expand it, and you end up getting, after 20 days, you start forming cerebral tissue, and you keep maturing it over a period of 40 to 60 days, we end up seeing a cerebral organoid. And the same thing now, if we were to do it from an induced pluripotent stem cells, we start with induced pluripotent stem cell colony, okay, and then we make embryoid body. The protocol is exactly the same, as you can see. It is not very different at all. And now if I were to compare a 20-day cerebral organoid to a brain, you can clearly see that in a 20-day cerebral organoid is going to closely match the mature brain of 6 months. Okay, this is basically second trimester of birth. That is what we are looking at. So this is close to around 60 days, I should say. So from 20 to 60 days, you can recapitulate the second trimester of pregnancy with respect to the fetal brain development. So the advantage of having an iPS derived cerebral organoid is that it doesn't necessarily have to be a very healthy individual. So if I want to understand a disease mechanism, a person has been afflicted with a particular disorder or a disease, I can take the skin of that person I can reprogram those skin cells into iPSCs, and then I can keep studying and seeing what has gone wrong during developmental stage in that person. And then we can come up with specific intervention therapy. So understanding basic biology is really important before we go on to do any kind of an intervention therapy. So I'm just going to play you a small video.
These researchers are using clusters of neural cells called brain organoids because they have many of the characteristics of a developing brain.
These are brain organoids. We call them brain organoids because they develop similarly as a human brain. We don't make these organoids by putting cells together. We just brain organoids. We call them brain organoids because they develop similarly as a human brain. We don't make these organoids by putting cells together. We just give cells the right conditions to grow, and they organize themselves into these structures. Like many stem cell researchers, we first start with induced pluripotent stem cells. These are made from a mature cell, such as a skin cell. The skin cells are given different factors, and they become pluripotent stem cells. To generate brain cells, we provide specific growth factors to guide the cells to become neuroprogenitors. These cells are the first step in brain development, and you can create all types of brain cells. First, these cells self-organize into neural rosettes, and we keep providing them with nutrients and factors for them to become healthy, mature neurons. As these rosettes grow larger, the cells form different layers that are similar to the ones found in the brain cortex.
These brain organoids are the focus of these researchers' many investigations.
Right. So that would have been something very striking for you when you looked at the video. The researcher actually showed that in a 6-well plate, he could have as many as 100 to 200 brain organoids. That is the power of scaling up. I mean, now imagine doing an experiment in 100 mice at the same time. You can't do that, right? So that is why we call this as a very powerful platform. And the number of starting material you need to make those brain organoids is only few thousand. So you can start with 9,000 cells and you can make as many brain organoids, okay? So now, if you want to see how these brain organoids look from within, you can clearly see that an immature organoid, the early organoid that we have, the day 15 or a day 20 organoid that we have, can actually have a lot of neural stem cells. So they are present as neural rosettes, which is what you saw in the video. And as they slowly mature, you end up getting a beautiful layer of these neuroepithelium, which gives rise to the whole neocortex. So you see that in red and green, you have multiple different types of cells there. Okay, so you have a completely beautiful layered neocortex that is formed in a more mature organoid. So this is what gives us the power to understand a lot of basic human biology. And the unique factor in these mature organoids is that you can see that there is on the right side, I have marked as outer subventricular, OSVZ, right? So these particular layers of cells have a specific cell type known as ORG, outer radial glia, which are very specific to humans. We are not able to find them in other species, including mice or in flies. So these outer radial glia cells are what we think underlie a lot of other differences that we see. So this way, it is quite interesting that even an immature organoid, not fully formed organoid, can give rise to inherently all these types of cells that we are seeing. And just the actual representation of what you saw in the cartoon in the previous slide, you have, this is immunostaining of a whole section of a brain organoid. So you can clearly see that you have neurons outside marked in green color. The progenitors from which all these neurons are going to differentiate are marked in red color. Okay, and all the other cells, because they've only stained for these two particular cell types here, but you also have all the other types like the glia, and you have the oligodendrocytes. All those other types are also present, which is marked in blue color. So blue color is the nuclei. So the complexity of brain is beautifully represented. I wouldn't say to 100%, but I would say that there is a close correlation of 85% at least we have in them. So it is not only about just having a single organoid, you also can start playing around with the types of organoids you have. So this is an example of a forebrain organoid or a cerebral organoid. So if you were to now give specific cocktails, we can also use specific patterning factors to make forebrain organoid. You can make a hindbrain organoid, you can make a midbrain organoid, all those you can do, and then you can make different types of brain. So this is forebrain. So similarly, you can make a hypothalamic organoid, you can make a midbrain organoid, different things you can do. And not only that, we can also fuse them together. So if I take a forebrain and a midbrain organoid, they tend to assemble together, okay, beautifully, and neurons can actually infiltrate from one part of the brain to another part of the brain. And by using axial patterning, we are also able to get different gradients across these organoids. So again, just a schematic representation of what I was talking before. You have forebrain, midbrain, and the hindbrain. And now if I were to take the forebrain, for just making a forebrain, I can give a particular combination of cocktails. For instance, if I want to make a dorsal patterning, just by using a combination of Wnt and BMP, I can just make a dorsal part of the brain. And by using sonic hedgehog or SHH, I can just make the ventral part of the brain. So like this, for each part of the brain, scientists have come up with beautiful cocktails which can be used to reliably get different parts of the brain. And then it's about mixing and matching. You can just try and see whether you can take the forebrain, midbrain, hindbrain fusions. So these we call as assembloids. Okay, so what it does when in comparison to a whole brain organoid is that in a whole brain organoid, you might not have all the representative pools of cells at the same time. Instead, if you were to make different parts of these organoids and fuse them, excuse me, so you can get a faithful representation of different types of cells which are present there. So what can we learn from brain organoids? So this is something which we have done here at CCMB. So we have made brain organoids using induced pluripotent stem cells. And in day 60 organoid, you can beautifully see that we have a dentate gyri, that is a part of the brain which we are showing here. And in green color, you can see that there are certain specific sensory neurons which have come in there. So what can we learn from these organoids? So of course, we can use it as a very powerful tool for disease modeling. Of course, we can use it to understand basic biology. And something which is a future outlook is that we can go for drug discovery. So in disease modeling today, I'm going to concentrate on how we use organoids for neurodevelopment, for neurodegeneration, for identifying cancers, and how we can use it to understand different infectious agents, how infectious agents specifically can cause particular perturbations in the brain architecture. So the first thing I'm going to talk about is how we can model fetal developmental diseases. So one of the rare genetic diseases that can occur is that a case called microcephaly, where small brain phenotype happens, okay? What happens is, like I said, 15 million neurons are born per hour during the first trimester. But when that does not happen, you have a smaller brain. So you can see a microcephalic brain has a much smaller layer of cells, thinner layer of cells. Therefore, the cerebral cortex size is drastically reduced. So there is a 2 to 3-fold standard deviation reduction in the head size. Obviously, this translates to a cognitive deficiency in these children as well. And this is what we call as microcephaly. It is a very rare occurrence. 1 in 10,000 children, 1 in 10,000 child is born with this kind of a phenotype. So like I said, Madeleine Lancaster was the first person who came up with the use of organoids for studying this microcephaly. So she used induced pluripotent stem cells from a patient who had a mutation in a particular gene because of which the brain size was smaller. Now, this mutation, when she introduced in mouse, she was not able to reproduce the microcephalic phenotype. So she knew that it was a very human specific trait that we are looking at. So then she took those iPSCs and she reprogrammed them into making cerebral organoids. And lo and behold, what she found was, in fact, that in brain organoids, she was able to reproduce small brain size. Okay, and when she closely start looking at the stem cell division from the red layer, so at the bottom of the panel, you see that in control, you have a beautiful red layer of cells. And then surrounding that, you have a huge, beautiful green color layer of neurons, right? This is what happens in controls. But if we were to look at the patient cells, you see that the neurons are all over the place and the neural stem cells are also completely distorted. They are not properly arranged at all. So there is a defect in stem cell division, and that is how they came up with the mechanism saying that in microcephaly, neural stem cell division seems to be affected, especially in this particular patient. Now, moving on to another rare type of a neurodevelopmental disorder, lissencephaly. I've already primed you with lissencephaly. I said that the mouse brain is already lissencephalic without having any kind of a folding, the gyri and the sulci. In fact, in rare cases, you also can have human babies who are born with this smooth brain phenotype, which is known as lissencephaly. These are very difficult to model because you can't model it in mice because mice already have lissencephalic phenotype. So where is it that you turn to? So people started turning to organoids. And when they took the induced pluripotent stem cells from this patient, and when they made organoids, they clearly could see that this patient who had lissencephaly had smaller and smoother brain organoids. And the reason they found out was the outer radial glia cells, okay, which are specific to humans, show a very slow mitosis in these patients. So this is what I was talking about, that having human organoids is a very powerful platform in case of human studies, because this study can never be recapitulated in a mice model, right? So they also, in addition to this, found that the cells showed increase in cell death and therefore they were becoming much smaller. So this is with respect to neurodevelopmental defects. So there was another type of neurodevelopmental defect where the connection of neurons between two different organoids, two different regions of the brain was affected. So this syndrome is called as a Timothy syndrome. And in Timothy syndrome, patients have epileptic bouts. Children have epileptic bouts. And how the scientists came up with a model for this is that they took cortical spheroid, okay, the cortex part, and they took the subpalatal plate, which is almost like the midbrain, and they fused it together to make an assembloid. So this fusion organoid you can see here in E, and they studied how neurons migrate from one part to another. So I'm going to play a small video for you. You can see that the neuron actually in control seems to move much faster compared to the neuron in case of the patient, which is moving much slower. So the intermigration of the neurons was very slow and it was very random. Okay, therefore these patients ended up having Timothy syndrome and they had epileptic seizures because the firing of the neurons was not uniform and it didn't take place properly. So this is again a very powerful way of studying not only a particular brain defect in one particular region of the brain, but also across different regions of the brain. And now moving on to other neurobehavioral aspects. Again, it's a neurodevelopmental defect, but a lot of you would have heard about autism spectral disorders. We have never been able to so far identify what exactly goes wrong with the brain of these children. And in comparison to microcephaly or lissencephaly, the thing about autism is that children often have macrocephaly, so they have enlarged brain sizes, right? So when people started making organoids from these patients, and they have done it over hundreds of organoids in hundreds of patients, and they have made organoids, they found that, in fact, these patients have macroencephaly. There is an enlargement of the brain size. And when they start looking at what exactly happens in these brain regions, you see that there is much more proliferation of these neural stem cells. So they are not organized as small layers. It seems to be that there is hyperplasia. So you see that the red layer in control cells is just a beautiful single bud, whereas in case of patients who have autism, you see a much bigger neuroepithelia. And you also see that the proliferation, which is marked by key 67 in red color on the right side, is much higher. The proliferation of these cells is way higher. So this is one reason why autism spectral disorder can result. And now what people are looking at is how do we reverse these phenotypes? Now, if we can make these patient iPSC-derived organoids and you add different drug combinations to see how they can reverse these roles, then probably we can come up with very good intervention therapies. And the next thing we can think of is neurodegenerative diseases. So we have seen neurodevelopmental, we have seen neurobehavioral or neurodevelopmental again, and here we are going to see neurodegenerative disease. So in Alzheimer's patients, we all know that people have dementia. It is a late onset disease. So in fact, what they found was when they took the patient samples and when they made brain organoids, in the middle panel, you see cleaved caspase 3, okay, that's marked in green color. And you see a drastic increase in the green colored dots in the cerebral organoids. So the Alzheimer's patients' organoids showed increase in apoptosis. Not only that, the synaptic connection between the two neurons was also drastically lost. And if I were to just show you a representation of these organoids, so if you see in 12-day, you have no difference basically. Again, in 16 days, across all three panels, you don't see any difference. 25 days, it looks fine. But over a period of time, in 40th day, if you see, the Alzheimer's disease patient organoids has become much smaller compared to the control or even the healthy control. So it means that there is a late onset of apoptosis which happens during Alzheimer's case. And they have attributed this to a particular protein called apolipoprotein E. And they show that by controlling the signaling cascade, this protein can actually cause an increase in apoptosis, especially during the late stages. So we've seen all the neurodevelopmental genetic diseases that can happen during brain development. Now, how about infectious agents? In fact, brain organoids has revolutionized our understanding of how neurotropic pathogens can attack the human brain. So a lot of you would have heard about Zika infection, Zika virus. So when mosquito bites, the mother actually becomes a carrier. She transmits it through the placenta, and Zika virus could infect the fetus, and then babies were born with a small brain phenotype. This was something which was massive in 2015 to 2017. It was a huge problem across the globe. And organoids proved to be a very powerful platform to study how exactly Zika virus could infect the human brain. They showed that in green color, you can see that the Zika virus specifically targets the human progenitors, okay, the neural stem cells which give rise to the whole brain. Now, if there is a depletion in that population, the brain size is going to be much smaller. And to give you a drastic example of what happens, you see that compared to the neurons which are in green color, the red colored layer is completely disorganized and you have huge apoptosis in this case. So this helped us come up with a molecular mechanism behind what could be the proteins, what could be the specific genes which are involved in this Zika replication. In fact, one of our studies showed that there was a critical RNA binding protein called Musashi, which could play a role in this Zika viral replication. So this was one of instrumental platforms in showing how Zika virus kills the progenitors. Now, in contrast to this Zika virus, if we were to now look at the other neurotropic virus called as the HSV, we clearly were able to see that even though HSV is causing microcephaly and it causes brain disorders, the way it infects the neural progenitors is completely different, right? And it was much more pathogenic than Zika. So in Zika, at least we can see some kind of a remnant cell architecture, whereas with HSV, it was completely catastrophic. Okay, and in comparison to another pathogen, torch pathogen, we call them, the maternal pathogen, which had a very mild effect. So you did not see a big cell death at all with respect to cytomegalovirus. So this is what we found out, and using these models, people were able to come up saying that why our body is able to better fight Zika virus compared to herpes simplex virus, because interferon beta in the host seems to be much more able to fight the Zika virus than it is to engage with the herpes simplex virus. So this seems to be the molecular mechanism behind why HSV is such a dangerous fetal pathogen. And we can't leave behind SARS-CoV too far because we are living in this pandemic era now. So the most recent study has in fact shown that SARS-CoV has the potential to target brain organoid. Specifically, they can enter the neurons and they can cause neurodegenerative pathologies, which can cause cell death. And more recently, from Madeline's group, they had a beautiful study where they showed how there was an entry of COVID-2. So they showed that there is a cerebrospinal fluid, which is around the choroid plexus, okay? And this pathogen SARS-CoV is able to breach this barrier. They are able to breach the barrier. They're able to cause leakage of cerebrospinal fluid and all the blood. So it's something like the blood-brain barrier, cerebrospinal barrier. So now the blood proteins leak into the cerebrospinal fluid, and that is how the whole brain has the potential to get infected. So these are, while at the same time they're enlightening, they're also pretty scary to look at. And next is, after infections, we look at how we can use organoids to study cancers. So there are multiple things which scientists have been doing. One, they have taken whole patient glioblastoma or the rare tumors which are present in children in the brain. They take those cells, because cancers also have stem cells, right? They also have stem cells. So they take that and they try to make them into an organoid by themselves. So they call them as glioblastoma organoids. And then they study what are the characteristic features of these organoids. The other way, if we want to identify for intervention therapies, and the other way, if you want to see how penetrant they are and how a particular gene can cause oncogenesis or not. So in the lab, if we identify a gene and if we want to know whether that gene is an oncogene or not, we can just take those embryonic stem cells to make organoids. We can just add these oncogenes to those embryonic stem cells and see whether these cells can themselves give rise to small tumors within the brain organoids. So this is again a powerful tool to study invasion and formation of glioblastoma. So an example on the right side I have given. So just for using two different genes, namely the by MYC overexpression alone, people have been able to show that these organoids can develop tumors. So I mean, in the next slide, I'm going to talk to you about what is it that it's not always positive about organoids. They do not have blood vessels in them. So what they did was they took the organoids and they implanted it in the mouse kidney capsule. So they would have blood vessels in them. And then they study how this invasion by different cancers take place. So you can see that at the bottom, there is a kidney capsule present on which they would graft the organoids. And once they graft the organoids, they can see whether tumor infiltrates or not and how big or small the tumors can be. And about the basic biology, what some of us are more inclined to is understanding how did our human brain evolve. Now, if I were to just take iPSCs from humans and chimp and gorilla great apes and make organoids, people have shown that in fact, inherently human cells have the potential to make bigger, better brain organoids. And this is again a cross-section showing a human brain organoid in the left side. In the middle, you have the gorilla organoid, and on the right side, you have the chimp organoid. So you can clearly see there is a size difference. And the way these tissues are organized are also very different. We still have a lot of neurogenic niches compared to chimpanzees, in fact. And the reason they say why this difference is there right from day 10, they say is that just a simple tweak in the cell shape during division. So on the left side, you see that the gorilla brain, actually the cells adopt a very cylindrical shape, which results in them migrating faster and depleting the neural progenitors. Whereas in human cells, we still maintain the cylindrical, this beautiful shape, not cylindrical, but I would say that this is this kind of asymmetric shape. And this therefore prevents the cells from prematurely differentiating. And they have even identified that there is a crucial gene called ZEP2, which is important for this cortical expansion. So thus organoids, it is not only useful for studying diseases, we can also use it for studying how is it that a brain has evolved, what is the difference between the great apes and us, and all those kind of basic fundamental questions also can be studied using these organoids as a tool. So what are the future directions that we look at? So like I said in a couple of slides before, the brain organoids as such, when we grow in the lab, do not have vasculature. They don't have blood vessels in them. So placenta, actually, you have all your blood and all the blood vessels are also formed. So one way scientists have come up with solving this problem is that they co-culture cells. So they take embryonic stem cells, iPS cells, and they mix it with the endothelial cells, which can give rise to whole organoids. The other way is that using microfluidics, they can make vascularized organoids in the chip. Okay, or you can just make all the blood vessels and you can forcefully express a particular gene. For instance, when they have this particular X-like transcription factor and they forcefully expressed it in embryonic stem cells, organoids could start forming blood vessels. So these kind of things are what people are doing to vascularize brain organoids, especially for cancer studies. That is very, very essential. And what are the other things which people can do? You can look at the neural and the non-neural interactions. So in fact, brain has its own immune system in spite of having the blood-brain barrier. So these cells are called as the microglia, which are inherent to our brain. So what we can do is we can introduce microglia, co-culture, forebrain organoids, or whatever organoids with microglia, and then with the blood-brain barrier, you can end up forming a beautiful whole brain altogether. Similarly, you can make a neuroendocrine axis. So you take multiple organoids like they did the forebrain and midbrain. You can now take the adrenal gland, you can take the pituitary, you can take the hypothalamus, and you can make these assembloids, which can give you a beautiful neuroendocrine axis. It is easily said than done. And similarly, now something which has been much more in focus is the gut-brain axis. So if we can fuse the vagus nerve with the intestinal organoid, then that would be really, really good, because you would be able to mimic the whole gut-brain axis, which is an interesting study. So what I've shown you so far is that using brain organoids, we can study neurodevelopmental disorder, neurodegenerative disorders, several disorders which underlie the basic molecular mechanism underlying mental healthness, toxicology, which I didn't speak about, where you can take these brain organoids and expose them to different maternal pathogens and maternal toxins which the placenta could face. In cancers, we can use them, and for infectious diseases, including a lot of viruses and bacteria. So these organoids, therefore, are a very powerful tool, and we can use them to understand how our brains function. So I hope I've convinced you of how powerful a platform this brain organoids are, and I'm happy to take any questions now.
Thanks, Dr. Pavithra, for the very interesting and insightful talk. So the way we usually do it is people have written several questions in the chat box, and I'll just read it out to you so that everyone can also hear what the questions are, and I'll just read out the questions and then you can just answer them. So there are several questions. I'll try to group some of the questions which are based on similar theme. So there are some questions on what are the similarities between the structure and types of neurons and glial cells and organoids versus an actual brain, and also in terms of the densities of different types of neurons and glial cells in organoids versus an actual brain. So if you could just comment on that.
Yeah, so I'll just go with that. So of course, we are not talking about the size differences you have to take into consideration. The brain organoids we make is typically 4.5 millimeters. At its maturity, it's probably the size of my thumbnail. So of course, the complexity of the structures, the number of cells stochastically is represented. There are few population of cells which are missing definitely, like the microglia are definitely missing from the brain. We have very few brains. That is the same case with certain sensory interneurons. sensory interneurons. So these kind of things, that is the reason why instead of making whole brain organoids, people are trying to come up with scaffolds to make bigger and better aerated organoids, because the thing with these organoids is that they start forming these clumps. You're working with a self-organized clump-like structure which does not have vasculature. Therefore, over a period of time, there is no oxygen supplied to the cell density. You end up seeing a lot of cell death. So that is one of the reasons why the cell density and the population might not be accurately reflected. But once we circumvent those problems, it should be doable.
Right. There's one question which is saying, as PNS is absent in organoid models, then how does it affect the neuronal connectivity and circuits?
What is
the peripheral nervous system is absent in organoid models, then how does it affect the neuronal connectivity and circuits?
Okay, so the neuronal connectivity and circuits we are talking about here are not from the peripheral nervous system. We are only talking about the CNS here. So PNS we are not talking about. That would be the last thing that I said, the neuroendocrine axis. When you get that, then probably we'll be able to get those.
Right. Something on similar lines. So someone is asking if the firing pattern or electrophysiology is, how does it compare to a native brain?
It is shown actually, there have been beautiful studies actually which have shown that the firing patterns are very, very comparable. So for this, people did not use the traditional organoids. They used something called as the air-liquid interface organoids. So you basically section the organoids and you expose them. So you have proper aeration as well as this, and then you add chemicals like calcium triggering and all that, and they find beautiful neuronal circuitries. So that has been done, and they do show that it is quite consistent. The electrophysiology part of it has been well established.
Right. There are two questions on whether it is possible to study disorders like schizophrenia or ADHD using these model systems.
So you can look at the developmental aspects definitely, like what people have been doing for autism. ADHD people have done, and also for schizophrenia people have done. I think a couple of studies are also in bioarchive. Behavior, these are not thinking, they are not conscious brains. That's something we should have in mind. There is no environmental input in that yet. So developmental aspect wise, yes, definitely you can study firing patterns, all that you can study, especially for intervention messages. But whether it's behaviorally relevant, I'm not sure.
Right. There's one question which is on, in case of developing disease models from somatic cells through iPSC technology, wouldn't the epigenetic fingerprint be lost? Does that have any influence on experiments and results?
Epigenetically and genetically, they are very, very, very similar. So this has been shown. There is a correlation of, I think, around 90-95%. So epigenetically and genetically, they're very similar. Imprinting wise, they're very similar.
Right. There's a question by Miron. Would it be able to study the effects of mitochondrial ROS and synaptic vesicles in Alzheimer's disease progression, like how we do with mice brain or post-mortem brains?
Definitely, compared to post-mortem brain, this would be much better. You can definitely do this. And I forgot her name, from University College London has been trying to do this, that they in fact have been trying to make these, the A-beta 42 aggregates. They have been studying all that in brain organoids. They have been able to do that, and they've shown it.
Right. There's one question. Can we study blood-brain barrier in organoids?
So blood-brain, okay, the forebrain or whatever organoids I spoke to you today about do not have blood-brain barrier. So what we have to do is we have to make blood-brain barrier organoids, so which is typically made using pericytes and using all the endothelial cells and a choroid plexus basically. So once we make the blood-brain barrier, you can fuse them with the forebrain, hidbrain, and the midbrain, and then that can be used. But a simpler way would be just make simple blood-brain barrier organoids, which people have done, and see penetrance of drugs, penetrance of viruses, how they leak, how they leach, all that can be studied.
Right. There's one question on what helps to maintain the progenitor or stem cells in the organoid environment, and how is that maintenance done? So there's one question.
So the cocktail of chemicals we typically add, like the fibroblast growth factor. There is EGF we add to it, and then we inhibit certain cell death using ROCK inhibitors. So there is a combination of different chemicals we use, and that is what inherently also puzzles people as to how is it that in a dish you're able to maintain this niche of neuroepithelial cells, like the neural stem cells, for a long period of time, which people have still not been able to find out. But of course, we supplement it with all the cocktails that are necessary to maintain the neural stem cells.
Right. There's one question. It says, is possible to maintain different organoids like brain organoids, liver organoids in a single plate, maybe for clinical trial purposes? I'm not sure if the person is talking about long-term maintenance or connecting different organoids.
Connecting. Okay, there is some ambitious project which is getting done from what I know about the gut and the neuro system I was talking about. Brain and gut, they are trying to go together here. Hans Kliver's group is trying to do that, which is very difficult because we are talking about multiple identities of tissues here. So if it is just the ectodermal lineages you're talking about, then yes, it makes more sense. Long term, people have been able to grow it for 6 months, or even some people say up to 9 months. These brain organoids have been grown.
Right. There are a few patients. Okay. So there's one question. There are few patients with brain tumors. Can we help them using brain organoids? Okay. I'm not sure what. I'm not sure if they're talking about transplanting these into actual human patients, if that's what the question refers to. So,
Okay.
yeah, I'm not sure what the question refers to. Okay, should be given. Okay, how to use. Okay, there's one question on how to use organoids to study immune interacting components, which involve several other cells in case of an organism, and how big a fused organoid would be if I have to replicate the whole immune compartment and lung. Okay. Maybe I think we can just compress it to ask, like, can we understand immune interactions in brain organoids?
Yeah, so you can co-culture. I think that was one of the slides I showed. You can co-culture the neuronal, the innate immune cells, or the system that you're talking about. You can co-culture them with the brain organoids, and therefore you can study the interaction. I don't think that is going to expand the size of the organoids. Whatever you do, it's going to be within the confines of that particular organoid. And we don't know what is it that makes the organoids, like probably the cranial architecture that we have serves as a scaffold to make it bigger. That is something which we have not been able to do. I mean, the maximum you can go to is probably the thumbnail size. That's what I said. Because after that, there is an onset of necrosis and we can't take care of anything else.
Right. I think similar on those lines. So there's like, how long can we keep an organoid alive? So in terms of number of days.
People have been able to do it 6 to 9 months, but there are some ambitious studies where they're trying to do it as long as possible, especially in the context of neurodegenerative disorders.
Right. So various organoids. Okay. So just a follow-up question on that. So in terms of the maturity of the brain, so even for the organoid which has been maintained for the longest duration of time, so what is the highest level of maturity that we have been able to achieve in terms of expression of various factors and structure?
So human age-wise, it is very difficult to say that, because for us, what happens is until until the 9th month, basically the fetus doesn't have too much of extra sensory stimuli and extra sensory inputs. So I think you can stretch it up to that. But after that, molecular mechanism wise, you can explore in the organoids, because once a child is born, then all the other factors come into play, which we can't provide in an organoid. So that is something which we also have to think very hard about, because for us, it is not only our genes, it is also the factor of environment which plays a role. So that is something which we have to think about.
Right. I think we can just take one last question, which is on how cost effective is this process? I think they want to compare it to the conventional process of using species. Yeah.
So the video would have shown you actually the reason why we think it is cost effective is because from 9,000 cells, you can get as many organoids. You can get, like, few hundreds of organoids you can get in one dish, small dish. The cost of feeding, I'm sure 1,000 mice is not going to be cheaper. And also, so those kind of things, yes, we have to think about.
Right. So there's, I think what Gayatri wants to answer, if you want to test cytotoxic drugs or chemotherapeutic drugs on organoids, and okay, I think there's one question on using organoids to see which drug works better.
Yes, that can be done. But this is where blood-brain barrier would be really important, because we can definitely go for the drug administration. I think there is somewhere in UK, I'm not sure which hospital it is, they have tried to use nano. So they did this trial in blood or brain organoids, and when they found out a particular regimen, they kind of just because it didn't penetrate the blood-brain barrier, they used a nanorobotic surgery to actually place the capsule of that particular drug with a slow release directly into the tumor, and they found that it did recede. So organoids are very powerful in that sense. Yes, you can check how cytotoxic they are and what is the therapeutic regimen that can be done with organoids.
Right. And I think there are one or two questions on interaction between various organoids, but I think we have kind of touched upon that. So we already covered that. So yeah. So I think we have covered almost most of the questions. So if there are any more questions, they can also, I guess, write you later on, or they can get they can reach out to us. So if there are any more questions, so yeah, I think there are no more questions. So thanks a lot, Dr. Pavithra, for giving such an interesting talk. So Sham, do you want to say anything? Do you want to end the meeting?
Thank you, Dr. Pavithra, for taking time and interacting with all of us on a Sunday morning. And also, I would like to thank all the participants who have joined us and engaged with Dr. Pavithra and asked many questions and clarified their doubts. So having said this, we hope that all the participants, we see you all in the next webinar soon. And for the sake of reference, we will be uploading this webinar video onto YouTube, and the link would be sent across to all the participants and the registered, all the registered participants. So if there's anything else which you would like to interact or would like to collaborate with us, please feel to write to Dr. Surat or myself or Dr. Brinda, and then we can take it forward. And thank you all for joining us, and hope to see you in the next webinar.
Yeah, thank you for being a very interactive audience. Thank you.
Yeah.
Thanks, Dr. Pavithra. Thank you, everyone.
Bye, everyone.
Bye. See you.